Hi everyone, welcome to yet another lecture series on overview and integration of cellular metabolism. We are discussing carbohydrate metabolism and today's lecture is on hexose monophosphate shunt. We will be learning about the steps and phases of hexose monophosphate shunt. Specifically, we will be covering the concept how you can classify the hexose monophosphate shunt pathway into the oxidative and non-oxidative phases. We will show how the regulation of HMP shunt is done and what is the metabolic and clinical significance of HMP shunt pathway. So, hexose monophosphate shunt, the short form being HMP, is known by many names. It can be denoted as PPP, which stands for pentose phosphate pathway, named after discoverer Warburg-Dickens pathway. It may be known as phosphogluconate pathway after a very important intermediate that is produced in the HMP shunt. So, mind it, if you see any one of these in any question, be it multiple choice question or be it any explanation downwards in the lecture session, mind it, we are referring to this one and the same thing of HMP shunt. So, what is HMP shunt? We have already learned about glycolysis and tricarboxylic acid cycle that does oxidation of glucose. Well, this is an alternative pathway to glycolysis and TCA cycle for oxidation of glucose. Mind it, the keywords alternative and oxidation of glucose. HMP shunt is anabolic in nature, again an important keyword because it will help you to understand the regulation of HMP shunt, and no direct ATP is produced or utilized in HMP shunt. This is also another important thing that we need to remember when we are discussing about HMP shunt. So, without proceeding further, from your lecture, from the lecture on enzyme regulation, will you be able to tell me, since it is anabolic, who or what will help in the uplifting the HMP shunt, or who will help in down regulation of HMP shunt? If you can, very good, write it down and we will see later whether your answer matches the explanation. It means you have paid attention to the earlier classes, right? So, the importance of hexose monophosphate shunt is, why do we need hexose monophosphate shunt? If you need to answer in one sentence, it is production of two very important compound, NADPH and pentose, specifically pentose sugars. Okay. So, NADPH pentose is the resultant of the HMP shunt pathway. Again, another MCQ thing, what is the total amount of glucose that enters this pathway per day? Roughly 10% of daily glucose. Where does the other glucose enter? Of course, through the other pathway, that is via glycolysis and TCA cycle, the main path, 90% goes there, here 10%. Next, RBC and liver, they metabolize more than everyone, 30% of glucose by this pathway. Okay. So, these are few facts that you need to remember, and these may prop up here and there as multiple choice questions. So, be mindful. Location, regarding the location of HMP shunt, a hexose monophosphate shunt, I may abbreviate as HMP, it is located in cytosol, means the enzymes for HMP shunt are present in the cytoplasm. Okay. And what are the tissues that are rich or that utilizes HMP shunt? Liver, fat cell adipose tissue, suprarenal gland or adrenal gland, RBC, testis, lactating mammary gland, they are highly active in HMP shunt. How this information will help you? Number one, you need to remember where these HMP shunt is happening, preferentially to other carbohydrate or glucose utilization pathway, and this can also be framed as an all except type of question. So, HMP shunt is highly active in all except. So, might be these tissues are given, okay, and brain is a third option or fourth option, you need to select that one. So, be very careful. So, these are the tissues in which HMP shunt is happening best, right. So, why it is important in these tissues? Because most of these tissues are involved in fatty acid biosynthesis, a topic that will be again covered later when we are dealing with lipid metabolism. It helps in production of fatty acid and steroid, biosynthesis of fatty acid and steroid, which is dependent on the supply of NADPH. And I told you, whenever we need NADPH, we need HMP shunt. Mind it, the major contributor of NADPH, that is almost 95% of NADPH of the body is from HMP shunt. So, where is the rest? 5%, just for MCQ. Mind it, it is malic enzyme. A very minor amount of NADPH is provided via malic enzyme, but that is close to negligible because 95%, more than 95% is formed from HMP shunt. Okay. So, this is the whole pathway where it starts with glucose 6 phosphate, where NADPH is produced. There are enzymes like glucose 6 phosphate dehydrogenase, epimerase, ketoisomerase, transketolase, transaldolase, and ultimately it lands into glucose 6 phosphate. And I just confused you, like, yes, it's a hell lot of intimidating if you just look at the overall pathway. So, this is not the right way to look at it, right? Yes, this picture or this diagram is present in all textbooks, okay, and it will get simpler as we go along, right? So, right way we can divide the whole thing into phases, okay. Considering the first 3 steps, these are known as the oxidative phase of HMP shunt. So, step 1, step 2 and step 3. So, let us see what is happening. See, this is glucose 6 phosphate, this is a cyclical structure of glucose which is acted upon by the enzyme G6PD or glucose 6 phosphate dehydrogenase. This is the short form. Okay. It utilizes NADP plus with the help of magnesium ion as a cofactor, converts NADP plus to NADPH plus H plus or reduced NADP or NADPH. Okay. Very important, and this is the rate limiting step or key regulating step by which the whole HMP shunt can be controlled, right, and it leads to the formation of the product is 6-phosphogluconolactone. Okay. Mind it, I told you gluconolactone pathway due to production of this. Okay. This is one very important intermediate after which the whole pathway can be named. In the next step, what happens? There is addition of one water molecule. Okay. So, one water molecule is added. So, simply HOH is added over here, OH is incorporated and here an H is incorporated, and over here we can see this is the carboxyl group has been added. So, when a compound ends with COO, it becomes an acid. So, this is 6-phosphogluconic acid. Let me pause here. If you are having difficulty in memorizing the structures, it is purely allowed. You can simply answer any question without knowing the structure, okay, and that is well and good if you can memorize it correctly. But if you are more interested, just by looking at the structure, the reactions can be better understood or it may appear more complicated. So, suit yourself, you can simply cover the structures and you can simply read out glucose 6 phosphate is converted to 6-phosphogluconate, it is converted to gluconic acid, like that, okay, well and good. So, next what happens? A dehydrogenase enzyme acts on 6-phosphogluconic acid. One molecule of carbon dioxide is lost, NADP plus is reduced to NADPH plus H plus, and ultimately since one carbon atom is lost, this 6 carbons, so you can see 1, 2, 3, 4, 5, 6. So, glucose is a 6 carbon compound, this is the okay, and it gets converted to a 5 carbon compound. The hexose is getting converted to a pentose. So, ribulose 5 phosphate is an example of a pentose sugar. Okay. So, NADPH and pentose both we have got. So, there is no need of studying the other part of course there is, because the cycle or the step does not end here, but here ends the oxidative phase. Why oxidation? Because there is loss of hydrogen. Where hydrogen is donated, hydrogen is accepted by NADPH. So, NADP plus is getting reduced, right, and the whole thing is getting oxidated. So, as and when NADP plus is getting converted to NADPH plus, dehydrogenation actually means oxidation. So, this is an oxidative phase. So, 2 dehydrogenases with an interim hydrolase phase. So, once this is formed, this is the ribulose 5 phosphate. This ribulose 5 phosphate gets interconverted. to various isomers. Okay. So, via an isomerase enzyme, this keto group ribulose 5-phosphate COO is a ketose sugar, it is a pentose, okay, ketopentose, it gets converted to a CHO. Simple, the formula is same. If you count the number of carbon, hydrogen, oxygen, phosphorus, they are absolutely same, just an aldose, I mean the keto group has been changed to a CH or aldehyde group. So, it is ribose 5-phosphate, extremely essential for synthesis of DNA and RNA, again a pentose. Or it can be altered in some other way where the terminal groups are not altered, but the intermittent groups are altered. You can see in this carbon it has been, the HOH has been altered. Mind it, any form of or any change where the HOH in the interim carbon is getting altered is known as epimerization, and these two isomers are known as epimer. Okay. So, ribulose 5-phosphate and xylulose 5-phosphate are epimers because HOH is changing in an intermediate carbon other than the end carbons, and the enzyme is epimerase. Basically, it is a special type of isomerase where there is an intramolecular shifting of bonds, right. So, next step is isomerization. So, after isomerization what happens? Very important, this ribulose and xylulose, right. So, both are 5 carbon, this ribulose and xylulose have 5 carbon. So, with the help of transketolase enzyme, what is happening? A 2 carbon is getting shifted from ribulose to xylulose, okay, and it is getting converted to pseudoheptulose, and this is converted to glyceraldehyde 3-phosphate. So, the end product both are 10 carbons. So, 5 plus 5, there is a 2 carbon shift and leading to the formation of a 7 carbon compound, that is pseudoheptulose 7-phosphate, and a 3 carbon compound glyceraldehyde 3-phosphate. Okay. Clear? So, next what happens? Again there is a shifting of carbon unit, a 3 carbon unit from pseudoheptulose 7-phosphate. Just for the sake of ease, I have excluded the structural layout. You can see 3 carbon unit is getting transferred from, so this is a transketolase reaction which I just explained in the last slide. Okay. So, transaldolase reaction, a 3 carbon is getting shifted from pseudoheptulose to glyceraldehyde. So, now 7 plus 3 becomes 4 plus 6. So, 3 from here is getting added to this 3, it becomes 6, and 3 is removed from 7, it becomes 4. So, what are the resultant compounds? They are fructose 6-phosphate and erythrose 4-phosphate. So, this is fructose 6-phosphate and erythrose 4-phosphate. Fine. You can again follow along very slowly, watch it again, rewind, it is up to you. Okay. But if you are clear up to here, next we can learn the second transketolase reaction. Very important thing, did you notice what was the cofactor in transketolase reaction? It is thiamine pyrophosphate, abbreviated as TPP. You have read in pyruvate dehydrogenase complex reaction, it is a form of vitamin B1 or thiamine. Okay. So, thiamine pyrophosphate TPP is a cofactor of transketolase reaction. So, again a transketolase reaction happens where a 2 carbon unit is transferred. So, 2 carbon unit is transferred from xylulose to erythrose, leading to the formation of fructose 6-phosphate and glyceraldehyde 3-phosphate. So, 5 plus 4. Okay. So, 2 gets converted, added here, it becomes 6, and 5 it becomes 3 carbon compound. So, ultimately glyceraldehyde 3-phosphate and fructose 6-phosphate, these two are produced at the end of the second transketolase reaction. Okay. So, you may ask where from the xylulose 5-phosphate is coming? Well, xylulose 5-phosphate is coming from here. Okay. It is an earlier product which is now reacting, which is now utilized in this second transketolase reaction. So if you are getting confused on how a single carbon atom can, or a single glucose molecule can actually lead to formation of ribulose and xylulose, and how the two intermediates are acting among themselves, well, you are actually on the right track, because in order to fully understand the HMP shunt, we need to consider it as a whole system where multiple carbon atoms are going into the HMP shunt and multiple products are coming out. Okay. So finally, in the regeneration phase, what happens? Two molecule of glyceraldehyde 3-phosphate that are formed in the earlier step, this one is getting converted or condensed. So two glyceraldehyde molecule, they condense to form fructose 6-phosphate and then converted to glucose 6-phosphate. So 2,3 carbon is condensed to form 6 carbon compound. Okay. So fructose 6-phosphate can be converted to glucose 6-phosphate just by the enzyme phosphohexoisomerase, and this fructose 6-phosphate is not only formed at the end, it was formed in earlier steps also, in transaldolase and the second transketolase also fructose 6-phosphate are formed. They also get converted to glucose 6-phosphate by the action of phosphohexoisomerase. Why it is written half? Because considering glyceraldehyde 3-phosphate as a 3 carbon compound, it can only lead to half fructose, but in reality 2 molecule of glyceraldehyde 3-phosphate condense to form 1 molecule of fructose 6-phosphate and eventually 1 molecule of glucose 6-phosphate. So what is the summary of the pathway? Considering 6 molecule of glucose, okay, that are 36 carbons, because hexose, one glucose molecule has got 6 carbon atoms. So the first 5 carbon atoms are lost as CO2 in the oxidative phase itself. So 36 carbon atoms, that is 6 molecule of glucose, we will get 6 molecules of carbon dioxide, 12 NADPH are produced, okay. And how many pentose are produced? It is very easy arithmetic. If you can subtract the total amount, the 5 carbon pentoses are interchanged in such a way. So mind it, pentoses are produced in between, okay. Here pentoses are produced, it is useful in synthesis of DNA and RNA, but pentoses are not the end product. The reaction does not end after a pentose is produced. It has to proceed in such a way so that again glucose 6-phosphate is regenerated, right. So considering 6 molecule, 5 molecules are lost as CO2, 5 pentose are formed, and if they are not utilized in formation of DNA and RNA, they will be readjusted in such a way, 6 pentoses, because 30 carbons, that ultimately 5 molecules of glucose are regenerated, or glucose 6-phosphate is regenerated. So if we look at the bigger picture, then you will be able to understand that 6 molecules went in, 5 molecules came out, and then one entire molecule of glucose has been as if one entire molecule of glucose has been burnt out to form carbon dioxide, okay, and 12 NADPH have been generated, but this is actually not the case. So if we just consider one molecule of glucose or glucose 6-phosphate, if you think that it is getting completely converted to 6 molecule of CO2, it is actually working hand in hand with 5 other molecule of glucose 6-phosphate that are also entering together, but are regenerated at the end of the reaction, okay. So this is the total picture. So in the oxidative phase itself, 12 molecules of NADPH are generated, 6 molecules of CO2 are eliminated, and the entire non-oxidative phase, and also pentoses are generated, right, and the entire non-oxidative phase in which multiple pentoses are generated are actually to regenerate the reactant, that is other 5 molecules of glucose 6-phosphate that are regenerated at the end of HMP shunt. Mind it, when we are considering fructose 6-phosphate to glucose 6-phosphate, it is actually going in the reverse direction of glycolysis, which have already been taught to you in gluconeogenesis, right, reversal of glycolysis is gluconeogenesis. So you already know that how fructose 6-phosphate can be converted to glucose 6-phosphate, it is a reversible enzyme phosphohexoisomerase, okay. So to conclude, the lecture has elaborately covered the oxidative and non-oxidative phases of HMP shunt pathway. In the next lecture, we will be looking at the regulation as well as physiological and clinical significance of HMP shunt. So, that is it for today. Please note the importance of the products of HMP shunt, that is NADPH and pentoses, which are highly essential for all biological systems. So, these are my references for the lecture. Thank you for your attention.